And he'll be talking about Microhylid frogs from Madagascar.
Two more people tonight. One more person. Come on. All right, I'm just going to start. Hi, my name is Mark. I am a researcher at the Zoological State Collection in Munich. And for the last 2 years, I've been working on disentangling cryptic species from Madagascar, specifically microhylids, using microcomputed tomography. And I kind of want to make a case for the idea of, well, we have next-gen sequencing already, but we need to move into next-gen morphology to complement that sequencing. So first, you heard yesterday from Miguel some nice fun facts about Madagascar. Here are a few more. So we have about 530 frog species estimated. That estimate ranges up to 700. Right now we have 300 species described, so that's only 60% of all of the estimated species, and that represents 10% of all of the frogs in the world. We have no other amphibians. And 99% of those, probably closer to like 99.99, we have 2 non-endemic species of frogs. We have 1 species that's found in Africa as well. So all of that diversity is pretty much unique to Madagascar. On the reptile side, we have an estimated 470 species, so about 50 candidates. 92% of those are endemic, and so the reptiles are not quite as impressive as the frogs, but still it's quite significant. And all of this on an island smaller than California. So the reason we have this huge gap between the number of described species and the number of estimated species is not because we haven't been to explore. We found these species, we've sequenced them, barcoded them, and found that they are unique. But why aren't they described yet? Well, 1, we have a limited workforce, a number of people working on the animals, and 2, most of them are cryptic species. So here, for instance, we have 2 frogs. This one on the left was recently described, Buphus ancarafensis. And these are cryptic species. If you take one and the other, and you put them each in one hand, and you compare them, you cannot tell the difference between them. It's physically not possible. So how is it described? Well, I know they don't look very similar in the pictures. I couldn't find any better ones. Well, first we had to identify them, and for that, the sequence data was taken, barcodes said, okay, these are significantly different. In fact, they're not even sister species. But if you look at their morphology, they are identical. So how do we tell them apart? Well, in tree frogs, we have this nice advantage that they like to call a lot. And so you can take the recordings and you can integrate that into your other analyses and find some good justification for the differences. But what if you're working with specimens that are already in a museum and don't have any call data? Or you're working with female specimens, or you had the bad luck of being in the field and it was dry season and the frogs weren't calling. Well, then you have to turn to another data type, and for that, one fantastic thing is to look at osteology in the frogs. So traditionally, osteological research was basically take the frog, take it apart, remove the skeleton, and then and study the bones independently. That's very destructive, very time-consuming, and you only get a skeleton without any of the context, so maybe it doesn't get articulated right. An alternative is to look at clearing and staining, and if you clear and stain, you reveal the bones and the cartilage, but again, it's very destructive and it's very time-consuming, which means that you can't use either of these techniques on the holotype of a species. So if you have just a few individuals, you're really wasting a huge amount of material by taking it apart like this. Well, now we have micro-CT, and I can take a frog, I put it in a little jar, I put it in a machine, I scan it for 20 minutes, I process it for 20 minutes or half an hour, and I get a beautiful 3D model, and then I go to the machine and I take the frog out, and it's still whole. Which means that we can do all of the osteological analysis that we used to do traditionally in a much shorter time frame and without destroying any of our specimens. But there are some drawbacks. So we discovered in this new species of frog that we're working on that the tips of the bones have lost their ossification. They've become cartilaginous. But micro-CT cannot render cartilage. There are no known stains to solve that problem. If anyone knows any, do let me know. But that problem persists, and so we still have to combine this a little bit with the traditional methods. We don't have to use that for every frog that we study. We also have problems where sometimes the skin is the same texture, the same density as the bones, and we have sometimes large food items in the stomach that really obscure the rest of the skeleton. Both of these problems can be solved post hoc by doing some careful segmenting, but I just wanted to highlight the fact that there are still drawbacks. We do still have to draw on some of the traditional knowledge. So I've been applying micro-CT to look at one radiation of frogs from Madagascar. We have about 70 or so described species from the Microhylid, all in 7 genera. We've got about 40 or 50 undescribed species from this subfamily, and it's hugely diverse. We have some of the smallest frogs in the world, that's a finger. So this is Stomphia, some of the smallest. We have the largest microhylids in the world, Platodontohyla and Platypelus. We have almost every ecological niche that you can think of. We have terrestrial frogs, arboreal frogs. In fact, the only sort of niches that you might think of that we're missing are flying frogs and swimming frogs. So a hugely diverse radiation of microhylids. And if you look at the osteology of these frogs, you see that it recapitulates that diversity in the external morphology. So we have these burrowing frogs with a highly specialized skull. We have the miniaturized Stomphia, and this is a miniaturized Anadonthyla. These are teeny tiny frogs that have very dwarfed bones. You see the arboreal form with its bifurcated terminal phalange. So all of that diversity, you could say, is visible at the genus level. But this is only a small subsample, and if you look below the genus level, you see again a huge level of diversity. So this is almost every species in the genus Rhombophrene, and you notice that, okay, Rhombophrene testudo, the slide we saw before, okay, it's very specialized in morphology relative to the rest of the group. So we have Rhombophrene minuta and Rhombophrene longicrus. These have extremely long legs. They're actually leapers. They're not burrowers at all. We have some other frogs that are more generalists, these two here. So we have this huge diversity. But one of the benefits of this huge diversity is that we can use it in the systematics and taxonomy of the frogs. So I looked very specifically at the osteology of a group called Rhombophrene ceratopalpabrosa, and until 2003 this had just one species, then it got one more, Coronata. And the trouble with this group is that it's all based around this one frog, described in 1974 by Guibet, and the holotype of that species looks like this. So some idiot, probably in Paris, decided that they would stain the holotype of the frog, and it ruined the specimen. The morphology is completely warped, the color is gone, so we can't really use the external pits of the frog. We don't have a sequence on the specimen, but you put that in a micro-CT machine, you get a beautiful skeleton out of it. So we can still use the osteology of the frogs, even though we've lost the other data. And so we use that, and we've already managed to describe one new species last year. We've got 2 more coming in December, and we're working on 3 or 4 more. So 2 examples of that are these. There's candidate species 1 and candidate species 2. You can just see here that there's a little bit of differences between these species. Okay, they're extremely closely related to ceratopalpabrosa. They're from the same radiation. But if you look at the shape of the hip, it's narrower in 2 than in 1 or in the original ceratopalpabrosa. You can look at the prootics here to see that they're more flared in some species than in others. And if you take that data set all together, you notice that actually there's a huge amount of data that you can get from the osteology of these frogs to try and solve their systematics. And we also did, within one species, we were able to do 5 specimens. We scanned all in a row, and then looked at their osteology and saw, okay, the variability within the species is quite low, and we can compare that to the holotype of the other species, and finally solve their systematics. And so we've used that so far to describe several. We've got 2 species already. We've got 2 more coming in December, as I said, and we've got a whole load more that are going to be coming soon. So this is fantastic. We can use osteology and micro-CT generated osteology to solve these species complexes and to reveal new species. And that's great. But where can we go from there? Well, my colleagues and I have been working on solving the systematics of this radiation, this subfamily. And so we have here, this is a new and very rough draft tree of the Microhylid. And this is based on all of the available sequenced gene data plus a few new ones. And we can take that and we can integrate it with the osteological data set that I've generated myself and other colleagues have generated. And we can take all of that together and we can suddenly study the evolution of these frogs in a completely new way. We can look at their, the origin of the hip shape in the group, or when and how the toes diversified. We can look at repeated origins of miniaturization in this genus and this genus and this genus. So we have all of these different options that we can suddenly study by looking at the osteology of the frogs together with their phylogeny. But there's also one other really exciting thing that we can do with these frogs, which is, I mentioned already with the stomach contents. Here, Rhombophrene testudo, its stomach is full of dirt, and it may be the very first instance of true geophagy in a frog. As far as I'm aware, there aren't any others that just eat dirt. We haven't yet analyzed that any further, but that's really exciting. And another really exciting thing is this guy. So we scanned this frog, looked at the stomach, and said, hey, that looks awfully like a spinal column. Is that a leg? Those are some bones. Yeah, that's a frog in the stomach of the frog. So we then, you can take the skeleton out of the frog and look at it, and yeah, that's actually a frog inside the stomach of this frog. So there's a whole new frontier of the ecology of the frogs that we can study just by looking at the micro-CT images of them. So yeah, there's a lot to do with that, and so I would like to thank my supervisor, Frank, at the Zoological State Collection, Miguel, who's been sort of co-supervising me, Anu, who's been a huge help in all of the work that we've been doing. I must thank my funders, the German Science Foundation, or the German Society for Herpetology and Herpetoculture, and also the Gans Fund for flying me here and letting me talk. So, thank you all, and if you have any questions, I'd be happy to answer them.
So you noted you had 5 individuals from one species, and you said that was enough to show that there wasn't a lot of variation within. Is that the magic number? I mean,
no,
yeah.
not by any means. It's actually, that's the largest sample size we have of any species from this. So permitting restrictions in Madagascar say you can't take out very many specimens. And so the limit we have basically is that species has 5. The most we have of any species is 16, and that's from multiple years of research done in the 1800s.
So how good do you feel if you only have 1 or 2 or 3?
I think 2 is a good number. 5 is a really good number, and as soon as you have sizes up to 10, you'll be able to do a huge amount. Yeah. Yes, sir?
When you were speaking of the cartilage,
yeah.
what types of histochemical treatment has been used on the material? It would seem to me that histochemical could be differentiated.
So the trouble is that I don't know of any stains that change the contrast. They have to be absorbed into the cartilage. And as far as I'm aware, nobody has yet found a stain that's absorbed into cartilage and renders the X-ray absorption of that tissue any differently. But I could be, yes? Hello. Iodine does not work. Iodine is absorbed by everything in the body except for cartilage. It's been tried. Also, phosphotungstic acid is not effective, as far as I'm aware. So the next step may be to go to phase contrast micro-CT. So phase contrast is developing really quickly, and it's superb. There's a group in Munich who've actually developed this, but it's still very premature. Yeah.
So I assume that you can use these CT scan renditions to diagnose a species in a publication. Is that true?
Yeah.
And then are those scans like deposited in a museum so that they can be compared or borrowed like a specimen could?
Right, so actually the best thing is that we can make 3D PDF models. We can embed them as supplementary material to our papers. So I can upload a 3D model of the frog, and then anyone who has access to the paper has access to or has access to the internet, has access to that model. And they can just open it at home in Adobe PDF Reader, and they can do what they want with the frog. At a larger scale, we're looking to make our own sort of repository of all of the models for pretty much everything that we produce, especially all the type specimens, because it's like a museum collection.
Available.
Yeah, sorry.
Thank you.